Imagine a world where we can cure genetic diseases, improve crops, and even engineer the traits of future generations. This isn't science fiction, it's CRISPR. It holds the power to rewrite the code of life itself, to correct errors in our DNA that have plagued humanity for millennia. CRISPR, short for clustered, regularly interspaced, short palindromic repeats, is a revolutionary gene editing tool discovered within the defense systems of bacteria. CRISPR allows us to target specific sequences within the vast library of the genetic code and edit them with remarkable accuracy. It's as if we've been handed a molecular scalpel capable of performing surgery on the very essence of life. The heart of CRISPR lies in its ability to locate and precisely cut DNA at predetermined locations. It's like having a pair of molecular scissors guided by a GPS system that navigates the intricate, twisted landscape of DNA. These scissors are actually a special enzyme called Cas9, and the GPS is a short RNA molecule, the guide RNA, designed to match a specific DNA sequence. Picture the DNA double helix, that elegant spiral staircase of genetic information. Now imagine the guide RNA molecule, this tiny scout, searching along the railing of that staircase, meticulously comparing each step, each base pair, to its own internal map. When it finds a perfect match, it signals the Cas9 enzyme, the molecular scissors, to get to work. The Cas9 enzyme, upon receiving the signal, springs into action, snipping the DNA strand with remarkable precision. This targeted cut is the key to CRISPR's power, creating a break in the DNA that triggers the cell's natural repair mechanisms. When a cell detects a break in its DNA, it immediately sets out to repair the damage. It has two primary ways to do this, each with its own implications for gene editing. The first is called non-homologous end joining, or NHEJ. This is the cell's quick-fix solution, essentially gluing the broken ends back together. While efficient, NHEJ can sometimes introduce small errors, inserting or deleting a few base pairs at the repair site. These errors might seem insignificant in the grand scheme of the genome, but they can be enough to disrupt the gene's function, effectively disabling it. This ability to disrupt genes is a powerful tool in itself, allowing scientists to study the role of specific genes in various biological processes. But CRISPR's repertoire goes beyond just gene disruption. It also offers a way to rewrite the genetic code through a process called homology-directed repair, or HDR. In HDR, instead of simply gluing the broken ends back together, the cell uses a template to guide the repair process. Scientists can provide this template in the form of a synthetic DNA sequence, essentially slipping in the desired genetic changes. The cell, none the wiser, uses this provided template to stitch the DNA back together, incorporating the new genetic information into its own genome. This ability to precisely insert or replace genetic sequences is what makes CRISPR such a game-changer. It allows scientists to correct genetic defects, introduce beneficial mutations, or even insert entirely new genes into an organism's genome. Imagine the possibilities correcting the single-letter misspelling in the human genome that causes sickle cell anemia, deleting the faulty gene responsible for cystic fibrosis, or even inserting a gene that confers resistance to HIV. These are not just futuristic fantasies, these are the very real possibilities that CRISPR brings to the table. CRISPR is more than just a gene editing tool, it's a testament to the power of human ingenuity, a shining example of our ability to unravel the complexities of the natural world and bend them to our will. But with great power comes great responsibility, and the ethical implications of this technology are as profound as its potential applications. The promise of CRISPR extends far beyond the realm of theoretical possibility. It has already begun to reshape the landscape of medicine, offering new hope for patients suffering from a wide range of genetic disorders. Take, for example, sickle cell anemia, a debilitating disease that affects millions worldwide, caused by a single-letter mutation in the gene that codes for hemoglobin. Sickle cell anemia transforms red blood cells from supple, oxygen-carrying discs into rigid, sickle-shaped cells that clog blood vessels, causing excruciating pain and organ damage. For decades, treatment options have been limited, often focusing on managing symptoms rather than addressing the root cause. But with CRISPR, we now have a tool that can potentially cure this devastating disease. In clinical trials, scientists are exploring the use of CRISPR to correct the faulty gene in patients' own blood stem cells, the precursors to all blood cells. These edited cells, now carrying the corrected genetic code, are then transplanted back into the patient, where they can produce healthy red blood cells, effectively curing the disease. And sickle cell anemia is just the tip of the iceberg. CRISPR is being investigated as a potential treatment for a wide range of genetic diseases, from cystic fibrosis, a life-limiting lung disease, to Huntington's disease, a fatal neurological disorder. In each case, the goal is the same: to correct the underlying genetic defect and offer patients a chance at a healthier life. But CRISPR's therapeutic potential goes beyond just correcting inherited genetic disorders. It's also being explored as a tool to fight cancer, one of humanity's oldest and most relentless foes. Cancer, in its many forms, is essentially a disease of uncontrolled cell growth, often driven by mutations in genes that regulate cell division and death. CRISPR offers a new weapon in our arsenal against this formidable opponent. Scientists are developing CRISPR-based therapies that can target and destroy cancer cells with remarkable precision, sparing healthy cells from the collateral damage often associated with traditional cancer treatments like chemotherapy and radiation. Imagine a world where cancer is no longer a death sentence but a manageable condition, treated with targeted therapies that eradicate tumors with minimal side effects. Beyond treating disease, CRISPR is poised to revolutionize agriculture, a field that has sustained humanity for millennia, but now faces unprecedented challenges in the face of a changing climate and a growing global population. CRISPR can enhance crops' nutritional value, making them more resistant to drought and salinity, and increasing their yield. The ability to edit the very essence of life, to rewrite the genetic code that underpins our existence, is a profound power, one that comes with immense responsibility. As we stand on the cusp of this genetic revolution, we must tread carefully, thoughtfully, considering the ethical implications of this transformative technology. One of the most pressing ethical concerns surrounding CRISPR is the potential for unintended consequences. The intricate web of life is a testament to the interconnectedness of all living things. And even seemingly small changes to an organism's genetic makeup can have ripple effects throughout the ecosystem. We must proceed with caution, recognizing that our understanding of genetics is still evolving and we cannot always predict the full ramifications of our interventions. The precautionary principle, the idea that we should err on the side of caution when dealing with complex systems we don't fully understand, should guide our actions as we venture further into the uncharted territory of gene editing. Another ethical concern is the potential for CRISPR to exacerbate existing social inequalities. Access to cutting-edge medical technologies has often been unevenly distributed, with the wealthy and privileged often reaping the benefits while the poor and marginalized are left behind. We must strive to ensure that the benefits of CRISPR are shared equitably, that this transformative technology is used to uplift all of humanity, not just a select few. International cooperation, open access to scientific knowledge, and a commitment to social justice are essential to ensure that CRISPR fulfills its promise of improving human health for all. Perhaps the most contentious ethical debate surrounding CRISPR centers on its potential application in human germline editing. The germline refers to the cells that pass genetic information from one generation to the next, such as sperm and eggs. Editing the human germline could potentially eliminate inherited diseases from a family line, sparing future generations from the suffering caused by these conditions. However, it also raises the specter of designer babies, children whose genetic makeup has been altered for non-medical reasons, perhaps to enhance certain traits like intelligence, athleticism, or physical appearance. The idea of engineering future generations raises profound ethical questions about human autonomy, the right of future individuals to inherit an unedited genome. It also raises concerns about unintended consequences that could reverberate through generations, potentially altering the human gene pool in unpredictable ways. The debate over human germline editing is complex and multifaceted, with no easy answers. It requires careful consideration of scientific evidence, ethical principles, and societal values. CRISPR is transforming our understanding of life itself. Its potential to solve global challenges is immense, but it comes with great responsibility. Are we ready for the CRISPR revolution? As we stand on the brink of this genetic revolution, we must proceed with caution, guided by principles of wisdom, compassion, and a deep respect for the sanctity of life. Let us use this newfound power wisely to create a healthier, more just and sustainable world for all.